Hi, welcome to the lecture on environmental adaptation of animals. Now on this second video on the diving, we are going on the mechanisms of how to maintain the body active in these long-lasting non-breathing periods. Okay, let's look on the oxygen capacity of the diving mammal to compare with the humans. So although the seal, the body weight is less than half, it has about the same as much oxygen in the body as the humans. But if we are comparing that how much oxygen they have per kilogram, that's about double. And what we can see that it's in a little bit different places. So in this aquatic mammal on the right, they have more oxygen stored in the blood and less in the lungs. So it's not only that having the more oxygen is a good thing, but having it in the right place. And therefore in the general, these diving birds and mammals have a large blood volume and high oxygen carrying capacity, meaning that they have plenty of hemoglobin. We will see a little bit these variation in a short while. Okay, let's look on different diving mammals to compare with the human. So in human, the oxygen store is about 30 milliliter of oxygen per kilogram. And then we have these otteric seals that are there on the coast and diving in the shallow waters. And what is the difference? Is that in the lungs, pretty much the same size, but they have more oxygen in the blood and more oxygen in the muscle. So they have better oxygen store. They might have even 2 times more oxygen in their body as we do. And then we have these posic seals, so these real seals, and some of them are diving very deep, like the Weddell seal or hooded seal, etc. And what is the difference? First of all, they have plenty of oxygen in the muscle, plenty in the blood, but you can see these yellow ones. These are actually very low. That means that they are not having larger lungs when they are diving. They are doing the opposite. So why these deep divers are not using the lungs? This has been tested with a video camera. So there's a bottlenose dolphin that is pressing button. And when it's pressed this button, then this apparatus is analyzing the excellent air. So how much oxygen there exists, how much carbon dioxide there exists, etc. And at the same time, when they're testing, you can see that this toric wall is pressed because there's so huge pressure under the water. And this been tested also that what happens inside a diving mammal. So in the harbor purpose, it's been tested that putting it in a very high pressure that is pretty much the same as in the deep of 100 meters, you can see that this lung volume is reduced. It's minimal on this high pressure because this toric wall is pressed and etc. And these kind of experiments you can't test it with wild animal that is living. So this harbor porpoise was found dead, but it was not. It was still in all the organs were in good condition, so they were able to test it. So, but anyway, they are not having a large respiratory organs during the diving. And if we would dive, we have much less flexible thoracic wall. So that's why if we could dive there in the 300 meters, there's not this kind of modulation of our body. But the idea is that, okay, actually, it's not a good thing. Because the air in these conducting airways is not used for respiration. Only those that oxygen that is in these alveoles. So you have these tubes. The oxygen there is not used, only the alveole. And then, of course, when you are diving deep, then there is this pressure. So you need more work to prevent the lung collapse. And then the fourth thing is that, okay, what we have in the atmosphere is 70% or even more nitrogen. So the nitrogen in the lungs is the one that is causing decompression sickness. So the diver sickness. We will discuss it a little later. But anyway, so that's why they are not having more respiratory volume. Okay, let's look on different divers. So over here we have a respiratory volume of different animals. First of all, there is a dolphin and a whale. Then there are four different seals and three different penguins. And to compare the human, we have maybe 7 milliliters per kilogram is the respiration volume. They all have more, but there is a huge difference. So these penguins, this respiratory volume is enormous, whereas these lions and the seals that are diving deeper, they don't have so huge respiratory volume. Then, what about the blood volume? The blood volume in human is about 71 milliliter per kilogram, as much as in the penguins and, for example, dolphins. But in these seals that are diving deeper, they have more than 2 times more blood in their body. And they also have high hemoglobin. So that means that these oxygen binding molecules, you have more binding molecules per liter of, or milliliter of, or deciliter of blood, but also you have then more blood volume. And that makes that, okay, this oxygen store in the circulation is quite large in these deep diving animals. But in the surface divers, like the penguins, they are quite pretty much the same. They are using this large respiratory volume. Then the muscle. How much muscle we have to compare the whole body weight is that actually we humans, we are athletic. Because we are not living in cold water, we don't need so much fat. So that amount of fat is so enormous in these seals, etc. So that's why they have much less muscle than we have. But the difference is that in these muscles there is plenty of more myoglobin. So that is the oxygen store used in the muscles. And from the previous videos, we remember that, okay, we have this hemoglobin and then the myoglobin, and this myoglobin is stealing the oxygen. And it's not giving it back to the hemoglobin, so that's why these muscles are using this oxygen store by themselves. Okay, does it work like this? So first of all, the oxygen store. So you have high blood oxygen carrying capacity, high blood volume, and high concentration of myoglobin. Or then in the penguins, they were having more respiratory volume. Then there's a variance in the oxygen capacity. So the mammals that are diving deeper or longer, they have larger oxygen store than those that are diving in the surface. Okay, case closed. No. The problem is that this oxygen store is not enough to support the oxygen use. So they calculated with the Weddell seal that, okay, there's a little bit less than 40 liters of oxygen, and if it's used 2 liters per minute in a rest, so you can last for 90 minutes. But it's diving for 1 hour and not resting. It's moving. So therefore the oxygen concentration should be several-fold more. It should have much higher oxygen store than this one. So that's why there's a discrepancy. This is not the way it works. And the solution for this question is the circulatory adjustment. It's been tested a long time ago. They've been adding contrast fluid in this descending portion of systemic aorta. This contrasting fluid is non-toxic fluid that is not passing X-rays through. So then you can see that where the circulation is flowing. And in normal air-breathing animal, the flow is fast in the cranial and caudal artery, so there's plenty of blood in the stomach and these intestine organs, as well as on the renal arteries. But then it's within a few seconds, it will also cause that, okay, actually this kidney is having plenty of this contrast fluid. So actually all our blood is flowing all the time in the kidneys. And then when it's diving or submerged, you can see that there is no so huge flow in versus in the upper picture, and this should be now pale. This is the kidney. Where is it? There is no blood flow in the kidneys at all. So it's closing down the kidneys. And let's look on different organs. With this kind of experiment, they've been testing that, okay, what is the blood flow per minute, per gram? And we have areas like the cerebrum cortex and cerebellum cortex. The circulation is maintained in the diving. Then we have areas where it's reduced. In the retina and in the lungs, there is still quite much blood flow. Okay, of course, this diving animal wants to see something. But then in the heart, this reduction of circulation is very drastic. So that means that, okay, either this heart is working anoxic or very deep hypoxic, or it's reducing the activity. And then the third version is that, okay, in the liver, in the pancreas, this is more than 90%. So actually this circulation is keysed. It's closed. There is no operation in the kidneys, in the pancreas, in the liver. And with that, we can remain the blood flow to these essential organs. Okay, but these are old school experiments. They were doing this way. So this is not voluntary diving. So it's anesthetic, but it's must be there on a place. And if it's freely moving, then there would be sympathetic nervous system, so adrenaline, etc., because there is more energy needed. And therefore, this free diving and this old school experiment might give different results. We will discuss this in this on the next video. And in these old experiments, they were focusing more that, okay, we have the circulation adjustment, so there's only 3 organs. We have lungs, we have heart, we have brains. And actually the lungs are not important, so there is a heart-brain machine. Okay, how we can support the brains? When we have the circulation going on in the brains, of course, this is needed to support the energy. And in the brains, quite a lot of the energy is used for maintaining the ion transportation. So we have ion channels, and they are not working if we are losing the ion gradients. So there must be always a lot of sodium outside and high potassium inside, etc. And for that we use this sodium potassium ATPase. And this must be then remained with this activity. But it's been also tested with isolated neurons and brain slice. So it's not only because there's more circulation. So in the lab mouse, this membrane potential is lost very fast. So within 1 minute already, if this drop is 20 millivolts, so it's from minus 80 millivolt, it's been depolarized to minus 60. Practically, this will affect that, okay, there's no neural activity at all. Whereas these hooded seal neurons, they are operating much better. And so therefore these neurons are hypoxia tolerant or one way or another. And at the moment it seems to be that these glial cells, so the support cells of neurons, are the key to support the energy for these neurons. But there are still plenty of open questions. Okay, what about then the bradycardia? So closing down the heart. So when we are reducing the circulation, there is less need for cardiac activity, and therefore we can reduce the blood flow with the lower heart rate. So it's analyzing that, okay, how many blood vessels are active or open, so then you can reduce the heart rate if there's not so many blood vessels active, so then you can remain the blood pressure quite constant. And this is minimizing, of course, the energy consumption in the cardiac muscle. So if you are reducing by 60% to 1/6 this heart rate. And at the same time, it's also needed because the coronary flow was reduced. So if it would work all the time in the same speed, that would cause damage cardiac tissue. It would be working in without energy, and that could cause that you have some mechanisms that are active there and to trying to survive, get these get these muscle cells to survive, but anyway they are not non-functioning, or they might work with anaerobic metabolism, and then could cause that, okay, then you have more lactate, etc. What about then on the involuntary diving? So in this experiment, they've been testing that, okay, how much oxygen, how much carbon dioxide, how much lactate we have there. And during this involuntary diving, there was an accumulation of lactate. At the same time, there is a reduction of oxygen concentration, accumulation of carbon dioxide. And then, after the diving, there's plenty of lactate in the circulation because these muscles are releasing the lactate to circulation. And so that means that, okay, then at least in the theory, in the involuntary diving, you are working with anaerobic metabolism. And this rate of oxygen depletion and the aerobic metabolism depends on different tissues. So first of all, in the muscles, it drops very fast. This is exponential. But in these other organs, this is a linear. And you can see that within 15 minutes there is no oxygen at all in the skeletal muscles, but in the respiration and in the blood there exists still. And you can see that this blue line, this respiration, so what happens in the lungs, there's still oxygen left, but it's not perhaps available for the blood, so that's why it's decreasing the slowest. And then if we are comparing that, okay, how the blood oxygen concentration, how the oxygen stores in the muscles are lowered, and when they start to accumulate the lactate, you can see that there's, although immediately when this oxygen is reduced, it's the muscles are already producing lactate. So they are not waiting before there is no oxygen available and then start the anaerobic energy metabolism. They are starting it immediately. Okay, then what happens on the lactate during the recovery is that the skeletal muscle is releasing it, and therefore in the circulation there is this huge boost of lactate after going back on the surface. And this lactate is metabolized, and that will lower, so it's not lasting forever, it goes down because the lactate is metabolized, but it will take a long time. So in this experiment, after 60 minutes, it's still higher than in the beginning. So that's why the animal must rest on the surface. Thank you.